Hello, welcome. Good afternoon, everyone in the Philippines and in the Asian region where we have the same time zone. Good evening or good morning to others in your part of the world. Welcome to our webinar entitled Pinoy Biotek For Us. This is going to be an English webinar, so stay tuned. I am Dr. Rhodora Romero-Aldemita, Executive Director of ISAAA Incorporated based in the Philippines, your moderator in today's webinar. The webinar is part of a series on an array of topics about modern and conventional agri-biotechnology applications and products in the Philippine context. Under the project Know the Science, Strengthening Biotech Links of ISAAA Inc., and funded by the Department of Agriculture Biotechnology Program Office. The Philippine Biotechnology Regulation, Adoption, Research, and Development is a model system in Southeast Asia. Public sector research using modern and conventional biotechnology tools is starting to deliver improved products for agriculture and food. ISAAA Inc. is holding this webinar to increase understanding and appreciation of Pinoy biotech products by the general public, to provide science-based information to target stakeholders for acceptance and adoption of Pinoy-developed biotechnologies, and to contribute to improving Philippine agriculture through biotechnology tools and products. In today's webinar, we will feature two technologies, one on organic fertilizer and another on fishery. So the webinar will run for 2 hours with lectures and discussion at the end. We are live in Facebook and YouTube channels of ISAAA. All microphones are on mute. Can we go to the next slide? And questions should be entered in the Q&A function only. The chat function will be used by the panelists and for important messages to the participants and to the panelists. The presentations and the Zoom recording will be posted at the ISAAA Inc. website and freely downloadable in a few days. So I hope I don't see any more questions on that. We also invite you to stay till the end of the webinar and respond to a post-webinar survey, and respondents will be given certificates. So to start, let me now introduce our first speaker. He is the president of the Visayas State University, or VSU. He's also a university professor at the same time. He obtained his doctorate degree from Nagoya University on bioreaction engineering. His research works on microbials and their applications in soil fertility has been recognized widely. Our multi-awarded speaker is the current president of the Asian Association of Agricultural Colleges and Universities and the president of the State Universities and Colleges, Association of Colleges of Agriculture in the Philippines. Friends and colleagues, let us welcome Dr. Edgardo Tulin for his talk on BioMEG. You have the floor now, Dr. Tulin.
Thank you, Dr. Aldemita. Okay, good afternoon, everyone. I'm proud that I'm invited here. It's a privilege to talk to a group of young and other scientists and other workers in agriculture. And I'm also glad that ISAAA Inc. has actually invited me to speak before this audience this afternoon. And I'm actually given the task to present the first topic. This is about a technology which we developed in the Philippine Root Crops Research and Training Center based at Visayas State University in Leyte, Philippines. And the title of the technology is BioMEG, which is a microbial inoculant. Now, allow me to tell you a story how we came up with this technology. So I titled this technology as a biostimulant for improved yield and nutritional quality of sweet potato and purple yam. There are a number of authors or researchers working on this project for the past 3 years, starting in I think it was in 2018 or 17. And then we go into the pot experiment, and then go into the field experiment to test our inoculants. So this has been a work by various researchers, and this is basically funded by the Department of Agriculture Biotechnology Program Office for the whole duration of these projects. Okay, so now what's the background of this technology, or how did we know about BioMEG? Okay. The concept of this work actually came about as a response to our strategy in conditioning soils, particularly degraded soils, which are actually abundant in many parts of the Philippines. So there are soils which are really unfit for agricultural production. So we try to make or develop technologies that could perhaps utilize these soils. These are the soils that no agricultural products or no agricultural commodities can be actually cultivated successfully. And these are also the soils that are actually on the background have immobilized elements in the soils that are not utilized by plants. So we came up with the idea that since there are these type of soils abundant in not only in our region, but also spread across the country, we tried to find natural microorganisms or indigenous microorganisms that could perhaps, the idea is as part of the soil conditioning process to release micronutrients which are immobilized in some of these soils, thus unavailable for plants. No? So we concentrated on the utilization of copper, iron, and zinc as our micronutrients that could be utilized or that could be dissolved or released by these microorganisms in their immobilized state into the soil, which could then take up by the plants. Okay, so during our screening process, we actually conducted soil samples as our samples for isolation of microorganisms in all parts of Region 8. And from these soils, we isolated the microorganism that's actually would fit for our purpose. So BioMEG basically is a microbial inoculant, which is a Bacillus megatherium as the active ingredient. So if you notice, our title is BioMEG. The MEG there is the megatherium, and bio is for just a name signify that it's actually an organism. No? So it works on the principle that the subsequent nutrient uptake by plants with the use of the product in the presence of macro fertilizers was found to increase yield and improve nutritional quality. So notice here that in the presence of macro fertilizers, so utilization or the dissolution of the micronutrients in the presence of macro fertilizers will increase the yield and improve nutritional quality. Now, if you notice, we focus on two crops. It's actually sweet potato and purple yam. So why these crops? Okay. Actually, I'm working based at the Philippine Root Crops Research and Training Center. And two of our major crops are actually sweet potato and purple yam, in addition to cassava and taro. So we use these two crops because we know that sweet potato is actually our superfood, if you will. And it is also a highly adaptable crop to any conditions. So, and it's also becoming popular these days in terms of its consumption. And purple yam, as this is also a high valued crop. So we try to use these two crops to demonstrate the capacity of our BioMEG to increase the yield and improve nutritional quality of these two crops. So the technology, in other words, only a trace amount of this BioMEG is applied for a few days after planting to effect the desired benefits. And that's the expected yield increase will in crop production will translate into a corresponding increase in income and provide a sustainable livelihood to farmers. So this is the background why we have in developing this technology, or the effect of developing this technology. So in essence, our Bacillus megatherium is actually a natural occurring organisms. It's indigenous to the soil, so it's not an introduced variety, but actually we isolated it from one of the soil in some areas in Leyte. And of course, as Bacillus is a rod shape, and it is a characteristic because it is a highly sporulating bacteria, which means that it forms spores. You can store the bacteria for a period of time and then revive it when you need to use it. So that's why it's very important. It's also fast growing, which is all very important as an indicator why it's an effective organic stimulant, no, or microbial stimulant. So there were 3 criteria that we use. Should be sporulating because you need to store them in a medium or in a solid medium before application. It's a fast growing, and of course the other one is it is able to solubilize copper, iron, and zinc in the soil that would render them available to plants. So that's the idea. So why iron, copper, and zinc? Of course, if you notice, these are the micronutrients that are needed by the body, the human body. So zinc, of course, is important for brain growth and also for growth. Iron and copper, these are also important micronutrients for our human metabolism. So they are not only important for plants, but they are also important, of course, for human. So the principle there is when they are being taken up by plants, then there is a corresponding increase in the growth or development of the plants, plus the nutritional content which are translocated into the plants and then stored in a form that is available in this case, because these are root crops, so they are stored in the roots, and when you eat the roots, increases the nutritional content of the roots, which are taken by humans for improved health. So that is the overall essence of why we are working on this, no? And it's the culture of production of this technology has also been worked out. It's not very, as I can see here, later on, it's also been worked out by this project to enable us to produce the technology in a kind of a cheaper version, or a technology that can also be that doesn't need so much facility for reproduction. So okay, so the feature of this technology is it has a low cost, because when we reproduce or when we multiply the microorganisms, we are using also indigenous materials that are considered waste. And for the production of the microorganisms, it's environment friendly because the raw material for producing the microorganism, if you mean the carbon source or the nitrogen source, and other raw materials used are actually also considered organic waste. And then, of course, it's organic based, and we found from our experiment that it improves yield, nutritional quality, and subsequently our income. So we use for production of this inoculant locally sourced material, and so we said that it's more sustainable in terms of producing this. We have conducted 2 field trials. Area is, however, less than 1 hectare, but the 2 field trials in the farm we conducted to test the effectivity of this inoculant. So this is how we apply this technology for sweet potato and purple yam. 2 weeks after planting, we apply the microbial inoculant. Of course, you apply also a complete fertilizer of 60-60-60 kilograms per hectare, and use 12 kilograms per hectare of the bioinoculant or the microbial inoculant only once. So notice that in all our experiments, you also need, even if we plant sweet potato or purple yam, you also need to apply complete fertilizer because that is, of course, necessary by the plants. And if you do that, then the plus the microbial inoculant that will really make the effectivity of the or the capacity of the inoculant to work better. No? In purple yam, 1 month after planting, and then 1 month after first application. So you have also 2 applications here, 6 kilograms per hectare twice. So essentially, you will have 12 kilograms per hectare for the whole duration of the planting. So way of application, it's also stated in our brochure and how you apply that one. Okay. So in terms of production costs, okay, the unit price from our estimation is just an estimate, but actually if you do economies of scale, this will perhaps still be lower. But based on a 5 kilogram dried product, your material cost and your utilities, you would have about 218 pesos per kilogram of the microbial inoculant. So notice here that the raw materials for producing are the following. You have the ipil leaves, the cassava peeling, and then you need an incubator, just an empty water container if you want to do it yourself. And then, but of course, you need to have a pure culture stock of the Bacillus megatherium as a something that you have really to you can avail of, no? And then other lab supplies, and then you need, of course, other things for sterilization of the substrates, and then we also also provide costs for electricity and water, and of course labor. So 218 pesos per kilogram by inoculant is the price that we obtain. So of course, as I said in the previous slides, it's really you only apply minimal amount to the plants. You don't need to apply like the complete fertilizer that you usually apply a large quantity. But this one, since this is for micronutrients, you don't need to apply in large quantity. Okay, so for our test results, you have your marketable yield, cost-benefit analysis, and of course, we studied in terms of nutritional value, anthocyanin content of the tubers. So in terms of marketable yield for sweet potato, on the left-hand side of this slide, you will notice that treatments with applied with complete fertilizer plus the microbial inoculant really yielded better compared to the plants with without fertilizer and with complete fertilizer only. So I would like to emphasize here that in addition to complete fertilizer, you really need an inoculant which is will improve the efficiency of the use of the complete fertilizer. It's in sweet potato. That's the effect. In the purple yam as well, it's the same kind of result, but the more pronounced result can be observed in sweet potato, although in purple yam, the tuber yield is also increased significantly with the addition of the inoculant plus complete fertilizer. So for the cost-benefit analysis, you will also notice that the net income of crops or of sweet potato applied with complete fertilizer and the inoculant is actually like 3 or 4 times higher than the one applied with complete fertilizer only and even without fertilizer. So we assume a yield of 9 tons in the respectively for the inoculant plus complete fertilizer with a population of 40,000, assuming the selling price of 30 pesos per kilogram of sweet potato. But actually in the market now, I think the selling price of sweet potato is higher than 30 pesos. No? So you could actually earn more. So in other words, that what it's telling us is with the inoculant plus complete fertilizer, you really have a higher net income. For purple yam, the same is true. There is a pronounced, of course, increase in your capacity to earn net income with purple yam in the presence of the inoculant as compared to the with fertilizer and without fertilizer and with complete fertilizer only. So this is the cost benefit analysis in terms of purple yam. Okay, in terms of if you take a look at if you base our analysis on the purple coloration of the tubers after harvests, you will notice that with the inoculant and with complete fertilizer in purple yam, there is so much intense purple coloration of the tuber as compared to NPK and without fertilizer, and also a more pronounced purple coloration with sweet potato. Actually, we use the purple variety here, but sometimes the purple variety also depends on the soil. But this time, given the one type of soil used in planting all these crops, we see the effect of the inoculant plus complete fertilizer in terms of the kind of tubers or kind of purple yam tubers that we obtain. Okay, so in a nutshell, the impact of this is since we're talking about global competitiveness, the use of this technology would significantly increase the production of sweet potato and purple yam, and this would provide an adequate supply of sweet potato and purple yam for the development of their local value chains and industries that will essentially provide sustainable livelihood or income opportunities to sweet potato and purple yam farmers, processors, and other stakeholders in the locality. So the generated products can also be exported given the rising popularity of sweet potato and purple yam worldwide, especially for purple yam, because there is really a high demand for purple yam, not only locally, but also abroad. No, in fact, we are always being asked when they can buy purple yam in a large amount for processing and for export. For food security, of course, is one of the technologies that our university is trying to come up with in response to or in addressing food security or sustainability. And I think production of sweet potato and purple yam for creating livelihood opportunities is also one very important activities that the university is doing at the moment. And what's more, the fact that it has also improved the nutritional quality of our sweet potato and purple yam, aside from their inherent capacity as nutritious superfoods, will also be addressing micronutrient deficiency, which is very important in addition to food security, also nutrition security. The value of the importance of micronutrient deficiency is also being addressed now by our government and also by our scientists in response to the concern on malnutrition, and also for infants or for growing children growing for brain or growth development. So we are also addressing here climate change. Sweet potato can also be very resilient. It's a resilient crop. In fact, sweet potato is highly adaptable to any conditions, even in soil without nutrients. But in a way, if you have this kind of technology, find that the soil is not so kind of supporting growth of plants, then you can enrich it with application of some soil conditioner or ameliorants, and one of these is our organic fertilizer. No? And that will also combat environmental protection, since we are actually not using here synthetic or chemical fertilizers in improving soil fertility, but actually the technology employs a useful microorganism responsible for restoring soil health. So that is really very important. And the fact that the raw material used in producing the microbial inoculant also comes from organic waste or from waste derived from other crops, the more that it also very safe and well, that we can utilize, then the waste can be utilized at the same time, can also be used as fertilizer, soil fertilizers. So to summarize, the technology, of course, work on the idea that we have the micronutrients being released by the soil through this microorganism with subsequent uptake by plants. And this actually, in the presence of the microfertilizers, it was found to increase yield and improve nutritional quality. So actually, the prior experiments for this one and micronutrient uptake, micronutrient dissolution has been done in our basic studies, as well as the screening and characterization of microorganisms, and we're preparing also publications to this effect. Now, so this is not presented here in detail, but these are under publications now, and the characterization of the microorganism and its other characteristics. No? And it's also very important, since we are aiming or targeting micronutrient here to address micronutrient deficiency. I think is also one kind of strategy where we can improve yield or production. So, of course, I'm aware that there are other many types of microbial inoculant, but please notice that this type of microbial inoculant is specific for micronutrients released in the soil as an aid for microbial utilization of or plant utilization of nutrients. So this is different from other sources of microbial inoculant because this actually targets on micronutrient. There are synthetic micronutrient conditioning in the soil, but I try to avoid synthetic because I know that is also in the long run, this may not be work, or this may not work effectively, or this may not be very sustainable in its effect. No? So what's more, if you have a corresponding increase in income, basically everyone is happy, our farmers are happy, and hopefully provide a sustainable livelihood for farmers to address community and will have community benefits. And we're trying to develop here technologies that are adapted to the community and which may not necessarily be very difficult to adapt. No? So we have developed IEC materials as part of our deliverables for this project. There is a Visayan short and long brochure, and there is an English short and long brochure. Of course, we have also conducted a training to this effect to our farmers in Region 8 and also some DA extension workers. In fact, we have also developed a field guidebook for BioMEG for sweet potato and purple yam production and a promotional video in a CD for this kind of technology. So this work was carried for 3-4 years, funded by the Department of Agriculture, the Biotechnology Program Office, and with the number of workers also working in this field. So I guess this is the whole technology package, and the idea here is we are actually requesting our students to apply this for other crops, not only sweet potato and purple yam, but also to other short-term crops, and the hope of improving yield and also utilization of soils that may not be seen fit for agricultural production, but hopefully in the presence of this one will actually be useful for cultivation of our very important. So thank you very much, and I hope that you have gained some understanding on this technology and also on the kind of topic that we are working to respond to improving soil health and soil fertility. Maraming salamat.
Thank you so much, Dr. Tulin. This is a very interesting work, and we see that there is really a hope that we can expand this not only in the Philippines, but also in other parts of the world. And I know that there'll be a lot of questions. So I hope Dr. Tulin, you will stay so that we'll have you respond to the question and answer. May we invite all of the participants to please enter your questions to the Q&A function so that we can discuss them at the end of the webinar. Thank you so much, Dr. Tulin. Let's give
You're
him
welcome. You're
a
welcome.
virtual round of applause.
Thank you.
Thank you. And now we move forward to the next speaker, who is Dr. Casiano Choresca. He is the chief of the Department of Agriculture Fisheries Biotechnology Center, stationed at Muñoz, Nueva Ecija. Yes. He is also the officer in charge of NFRDI Brackish Water Fisheries Research and Development Center in Lanao del Norte, and the Career Scientist 1 at NFRDI. His current researches are in biotechnological approaches in the production of mudfish, induced breeding of climbing perch using induced spawning hormones, and promoting aquaculture in freshwater fishes. He also worked on molecular detection of parasites and bacterial diseases, emerging viral diseases in fish, including the tilapia lake virus and iridovirus in milkfish. He obtained his PhD in veterinary medicine at the Seoul National University, South Korea. Let us welcome Dr. Choresca for his talk on spawning technology for mudfish. You have the floor now, Dr. Choresca.
Hello, good afternoon. Good afternoon, Dr. Ola.
Good afternoon.
Good afternoon, good morning to our participants. So let me share my slide. Okay, so this afternoon, I'm going to share with you the induced spawning technology in mudfish. So actually, this technology has been used in different countries. However, in the Philippines, I believe that is the first effort here in aquaculture technology, the induced spawning of our mudfish. And this project is funded by the DE Biotechnology Program Office. And to this date, our project is on the infant stage, and we are now optimizing our protocols for the induced breeding. However, there are already experiments that have been done, and lately we have been on training of our stakeholders in the Philippines. So this is the presentation outline of my presentation this afternoon. So defining induced spawning in fish, and why is induced spawning essential to aquaculture, the spawning process in fish, so in a simplistic model, and then the science behind induced spawning, and the application of induced spawning technology for mudfish production, and some of our conclusions on this project. So induced spawning is defined as the regulation of environmental condition or by introduction of hormones to stimulate factors that trigger reproduction. And this is for the promotion of gonadal ripening and timely release of sperms and eggs. So in fish or in reproduction in fishes, it is regulated by external environment factors that trigger internal mechanisms into action. And the final event of the reproductive cycle is the release of eggs and sperm, resulting in spawning, and can be controlled by either placing the fish in an appropriate environment or by changing the fish internal regulating factors with injected hormones or other substances. And the internal mechanisms that regulate spawning are similar for most fishes. Actually, there have been efforts using the technology, especially in catfish, and this method, we modified it into mudfish or the snakeheads or our Chana striata or our dalag in the Philippines. So in many fish, fish spawn in environments that are nearly impossible to stimulate in a hatchery. So hormone-induced spawning is the only reliable method to induce reproduction in this fish. So why, sorry. So why induced spawning is essential to aquaculture? So this technique is very simple and does not need too much technical assistance or knowledge. So, and it removes uncertainties or breeding spawning. So usually you can control and you can collect eggs and you can control, of course, the reproduction of our fish. And the targeted schedule spawning is possible and can produce fry outside spawning season for hatchery and or grow out. So using in the spawning, you can control and you can get any time if your brood stock are matured. So they can release eggs and you can get eggs anytime for your experiments and or for your production. So for example, the reproduction cycle of mudfish, it matures after 1 or 2 years and it spawns in freshwater habitats, and spawning occurs throughout the year with its peak prior to or with the onset of monsoons or between February to March, June to September and November. So by doing induced breeding, you can control spawning and you can have readily available fingerlings anytime. So you can also provide pure spawn of fish under cultivation. So you can have a breeder selection is possible. You can have your good quality fish can be selected and bred. And you can have a higher chance that the offspring inherits the good traits of its parents. So you can have also your pure spawn and can be obtained from the fish under cultivation or under aquaculture conditions. So lastly, it offers more controlled hatchery or aquaculture operations. So anytime you can have in a controlled conditions, actually here in Fisheries Biotech, we have our backyard hatchery. It's a small scale hatchery and it's producing already our fingerlings and fry for our farmers. So the spawning process invests in a simplistic model. So why in the spawning in mudfish, and what are the mechanisms behind this? So environmental factors have been shown to play a significant role in the reproductive cycle in fish. And these environmental factors, of course, are photoperiod, water temperature, water quality, and weather cycles, spawning substrates, nutrition, and others. So these factors do not function independently of one another, but are interrelated. So while proper environmental conditions stimulate the reproductive process, any unsuitable conditions can immediately override any attempt at induced spawning. So this is the reason why it is quite difficult to stimulate natural spawning in hatchery conditions. So the internal mechanism that regulates the process of reproduction in fish is the brain or the hypothalamus pituitary gonad chain. This mechanism is complex and additional scientific information is continually being added. So the following is a simplistic model of this process. So for the environmental stimuli that are received and translated by the brain, stimuli of reproductive importance are routed to a portion of the brain called the hypothalamus. So the hypothalamus produces gonadotropin-releasing hormone, or the GnRH, and also gonadotropin-releasing inhibiting factors. So experimental results suggest that dopamine is a substance that inhibits the release of the gonadotropin. So the gonadotropin-releasing hormones, or our GnRH, is thought to stimulate the pituitary in a small gland located beneath the brain to produce and release gonadotropin hormones, or the GTH. So gonadotropin hormones, or our GTH, act on the ovaries and testes. And testes are in the gonads of the fish. So steroids and prostaglandins appear to be in the local ovarian mediators of GTH action and cause release of the eggs. And elevated blood levels of GTH trigger two distinct ovarian process. So this ovarian process are for evenly stimulated by prostaglandin, and the steroids also appear to induce spermation in the male. So in view of the aquaculture, so the natural spawning is difficult to control and predict our spawning, and there is an erratic production, and aquaculture operations are more difficult to manage. So by doing this process, it can ease production of our fish. So what's the science behind the induced spawning? So the objective is to bypass parts of the brain, hypothalamus, so the pituitary gonad sequence to speed up or induce spawning in the fish. So first is to regulate the conditions or the senses to stimulate in the hatchery the ideal induced spawning conditions of fish is to encourage spawning breeders. So like, for example, temperature, water conditions, feeds, and other factors. So the introduction of synthetic gonadotropin or GTH is to release hormones called gonadotropin releasing hormones, analogs, which directly instruct the fish pituitaries to produce gonadotropin hormones. So the introduction of the pituitary extracts or purified gonadotropins is to speed up the gonadal maturation. And of course, the introduction of the synthetic steroids or prostaglandins to trigger final maturations of oocytes and sperms. So there are already commercial hormones in the market now. So actually these are in this picture, the gonadotropin releasing hormone analogs. So this is not implies the the marketing and other production of these hormones. However, these are the common hormones that are being used in our laboratory, and these are the steroids and the prostaglandins, and the pituitary extracts or purified gonadotropins. So actually, one of our projects or one of our output here in this project is to develop purified pituitary extracts from fish. And this is an ongoing project from the Biotech Program and being implemented by ED4DI. So here's the process, the regulations of this hormone. So I have mentioned this on the mechanism. So these are the gonadotropin releasing hormone analogs that we have, like ovulin. I think we have the HCG and the likes. And of course, for the steroids, we have tested some oxytocin. Oxytocin are being applied in induced in dogs and in cats. Then the application of this induced spawning technology for mudfish production. So first, mudfish. So we usually call this in the Philippines haruan or haluan in other in other regions. So scientifically, this is called Chana striata. So maybe in other countries, this is called snakeheads. And here in the Philippines, we usually call that mudfish. So in our native language, we call that in Ilonggo as haruan or haluan. And the local name here in the Philippines is dalag. And here are the commodities that being produced here in the Philippines. So actually, I was in Mindanao last month, and there are lots of, like, for example, there's fermented product from the mudfish, and the cost of this fish is quite high in the market. So why this fish? Because it is white, firm, almost bone-free flesh and good taste. And according to the elders, it has a high nutritional value. and medicinal and pharmaceutical purposes. Actually, the soup is given to, according to the traditional practice, the soup is given to the newly birth women in the Philippines. No? So, this is air-breathing freshwater fish, and it has high aquaculture potential. Actually, in other countries, they are already farming this kind of fish. And in the Philippines, the stocks are dwindling, and that's why this is our efforts for the production using this intervention on induced spawning. And this fish has high tolerance to adverse environmental conditions and high profits. So actually, in the Philippines, this highly marketable in inland areas. Actually, here in our workplace in Central Luzon, Ilocos, Cagayan region, Cotabato, various areas in the Mindanao region. So I have said they have that fermented mudfish, no, that they sell in a high price actually in their market in Mindanao, and typically sold to live local markets and commercial sold as a fermented rice fish, typically called as burog dalag. So this burog dalag is called fermented fish, no, or fermented mudfish, as you can see in this picture. So here are some tips on our induced spawning process. So this is part of our technology, and I cannot disclose some of the key points of this project because this project is ongoing for publication and for patent. And this is the samplings done once or 2 months, and we measure and record the length of the fish, and we determine the oocyte maturity, and we usually treat the disease. Usually for parasites, we just get or collect parasites manually, and we have to do salt bath for fish. And the project aims to have or to select good quality and mature broodstock to be induced. So for sex identification, so usually male breeders have this lean, usually longer than females, but there are distinctive factors like some males they don't have long bulging abdomen, and the genital papilla is small, and anal papilla extra is prominent and the slit is pointed, as you can see in this slide. So the female, so actually female breeders distinguishing factors are quite distinctive, and you can see the soft and bulging of the abdomen, as you can see on this slide, and the genital papilla, so here, is prominent and round, and reddish protruding swollen vent, and the anal papilla-like structure is broad and slightly reddish, and T-plant with a reddish dot, so very distinct, so you can easily be recognized if it's female or male. So for the sexual maturity, so the ratio of the water to anesthesia is usually we have this induced spawning. No, we usually first we usually anesthetize the fish for induced breeding, and we check using cannulation, no? And it is done during sampling. And cannulation makes us easily identify the male to female. No? So this is how we do our sampling for the sexual maturity of the fish, as you can see in this slide. So for hormone injection, so we usually weigh our fish, and we determine the weight of the fish and the injection amount of the hormones to be injected. And for fish, we usually cover them with white cloth and to make it calm. So actually, this process has been already a practice in our lab. So this is to make the fish calm. I mean, especially this mudfish. And using the sterile syringe, we usually inject hormones, no, according to the weight of the fish. So as it been shown in this picture. And for injection, so we usually deliver in the above the lateral line of the fish here. And after injection, so gently we do some gentle massage, you know, in order to evenly um, deliver the hormones. And usually we place them in HAPA nets. So the ratio is 1:1 based on our practice to 1:1 fine mesh nets. So we usually place them in white mesh nets because in order to escape in order to prevent the escape of of some eggs if they're already spawning in the net cages. So for egg collection, we usually identify the eggs. So we usually collect the eggs. So the spawning occurs between 24 to 30 hours. So this is already optimized in the lab. So this is based on the hormone of injection. So you can get after 24 hours or less sometimes. However, usually this is the collection time that we had after 24 to 32 hours. And then you can see that the egg is already fertilized and it is non-adhesive and it is transparent. Usually the color is yellow. So you can really identify the eggs floating on in your net cages or in your tanks. And after we usually collect those eggs in between of 2 to 3 hours after spawning. So for incubation and hatching of the fish, so we usually incubate the eggs in basins and transferred to our fiberglass tanks. And usually for the months of December, going to December, November, December to January to February, we usually place some heaters because of the temperature. And the temperature ranges to 26 to 30 degrees Celsius. And also hatching occurs for 24 to 32 hours after fertilization. And then usually we collect eggs and we remove breeders and bring it back to their HAPA net cages. So for larval rearing, actually, we usually incubate our larvae in fiberglass tanks, customized fiberglass tanks, and with a stocking density of 10 to 15 individuals per liter. And we feed them with zooplankters or moina. Actually, in the lab, we have our purified moina. And we usually collect some mixed zooplankters from the ponds and give them to our larvae. And we feed them three to four times daily. So because they are still larvae and they need zooplankters to feed. And we feed them until 14 days. And then after 2 weeks, we usually place them in hapanate cages in ponds. So recently, we just modify these techniques in order to induce some growth in larval rearing and in order to also to minimize some costs. So this is also part of our experiments in the lag hatchery system or in mudfish hatchery system. So for stocking in nursery rearing, so based in our experiments, so we have this for 300 fry per square meter in hapanet cages. And we have tried some of the feeds like the trash fish, and we feed them for 2-3 times. And once a week, I mean, we grade the stocks and remove the shooters or other dead fish. And in conclusion, So Indus spawning technology in fish offers a straightforward, smart, and efficient approach to increase fisheries and aquaculture production, resulting to higher yields and increased profits to our fish farmers. And then this technique is easy to perform and does not require expensive materials or equipment. And induced spawning also helps addressing concern in food security, reduces dependence on our wild stocks, as I've said before, that this fish is already dwindling, and alleviates the sharp decline of fish population due to overfishing or anthropogenic activities. So here are our research team in this project and our collaborators from DFAR and from Mindanao State University in Naawan campus. And recently, we had undergone some induced spawning trainings here in our center, in Fisheries Biotech Center, both technical and research staff, and also our fish farmers for the promotion of this technology. And in Western Visayas, we have conducted already different trainings in Iloilo, Guimaras, and in Bacolod. And of course, product of our induced breed fish. So we usually give them to our fish farmers for aquaculture. And this is our research team, the Fisheries Biotechnology Center team. So thank you very much, and thank you very much for inviting us, of course, from ISAAA Incorporated. Thank you.
Thank you so much, Dr. Cass. Those are very comprehensive. That's a very comprehensive presentation. And I know that there are a lot of questions already coming in. And let's give Dr. Chas a big round of applause, virtual applause, everyone. Thank you so much, Cass, Dr. Cass. Now we will proceed with the Q&A. We have 30 minutes to do this. And I'm happy that Dr. Tulin is still with us. Can you spotlight on Dr. Tulin as well so that we can start the Q&A session? Now let's look at the questions while we're waiting for the questions for Dr. Cass. Actually, maybe you could just, Dr. Tulin, you can just respond this one after the other. But the important thing is, where is the how do you call this? The bacillus? Where is the microbe? Yeah, Bacillus vegetarium being isolated. What is its habitat? Is it endogenous or is it introduced? That's one. And then the second one is its application in other crops. Have you tried working on other crops besides potato? So one suggestion here is on cotton. And another one is, will BioMEG interact with herbicides or pesticides applied to soil or crops? And how about the other soil conditioning? And there's a very long question here from Grace Glenn Gabisay. So if you would, if the number of times that BioMEG product is applied for 12 kilogram per hectare rate, the schedule of application, the total amount, the duration, the mode of application, whether basal or side dress, method of application, whether foliar or drench, and lastly, this is biological product. So has there been no sign of toxicity, phytotoxicity, or deficiency there? And then we wait for the other questions for Dr. Cass. Those are many questions. So
Sorry, I can't remember all of this, but I will answer as far as I can remember the questions. But anyway, okay. Right. BioMEG is an indigenous microorganisms actually isolated naturally from the soil. No? So it's not introduced, it's a naturally occurring organism as bacillus. It's actually, the origin is from a soil that is not an ordinary soil, but a soil that is kind of from a previously utilized soil that has been kind of used for other things, no? And in other parts of Leyte. And what's the next one? Okay. The method of application, of course, is it's basal. It's the dressing in the soil. You apply it in the soil. The small amount is enough to get the desired effect of the inoculant, because it's actually, just as a name for it, induces or it stimulates the soil environment, no, and utilizing available micronutrients to work hand in hand with our macro fertilizers, our NPK, which are also applied in the soil. Mm.
Mhm.
What's the other question?
Have you
Mm,
tried it in other crops besides?
okay. Uh, we have graduate students, or we have undergraduate students now working on vegetable crops. We have yet to see the results this second semester, but I think it actually shows promise, because somehow there is, I know, but I cannot say for sure as of now, but there is a plan that we expand this to test for other crops as well, not only for sweet potato and yam. No, because I believe that since that will act as a kind of stimulant for soil microorganisms, it will apply also to the other crops.
And does it have a counter effect with other foliar applications, herbicides?
As far as that concern, we have no data yet. I cannot say for sure that it will counteract with a herbicide or a pesticide being applied, because we don't have that experiment yet. That's an interesting question. I think we can look at it perhaps if there is a kind of counter effect with the inoculant. But my guess is, since it's in the soil, it's a naturally occurring microorganism, it may persist in the soil as well without really being affected by pesticide or by other sources or by other chemicals. That's only my guess, but we have to do some experiments on that.
Okay. And it doesn't release any other biological products?
No, I
Or
don't think
by,
so.
yeah,
Yeah, I don't think so, because it's a pure isolate. It's a cultured isolate, and it may associate with other microorganisms, but in a more symbiotic way, because there are many microorganisms in soil. But the thing is, the major effect of that is actually, since it's already integrated in the soil by way of applying it in the soil, then it may actually make the other microorganisms in the soil work together with that microorganism.
okay. Yeah. So stay put for a while. We will
Yes,
go
yes.
with Dr. Casiano's questions. So Dr. Cas, on DAFBC, have you tried the biofloc technology as one of the biotech application in fisheries? If yes, may we know what commodity it is? If none, can you share your experiences on it? Biofloc, I have no idea what biofloc is.
Actually, Doc Ola, biofloc is containing those microorganisms in one in a culture system. Actually, it's quite related to the work of Dr. Tulin, but it's in biofloc systems in fish, or I mean in water. So in our case, we don't practice yet. However, in shrimp, there are already reports working on it. Like, for example, maybe I will cite here the work of Dr. Christopher Kaipang in University of San Agustin and now in UP Visayas, and that is also funded by the DOST. But in fish, we did not practice yet those here in FBC. But however, we have some experimental work, in a tabletop work using in the larvae, but still using I mean more on experimentation now, but not yet in full blast for fish, because I think we also have that in a small environment because of the characteristic of also of mudfish or our dalag, to feed them in our practice here in climbing perch. We use that in green water culture system. So we collect tilapia water system, and then we place our larvae Kasi what tilapia water system have that many microbial components, and it enhances also the water it acts also as bioremediator in fish. So that's our experiments, and maybe I can disclose, but we have already data here that it increases the survival of our climbing perch. So for the biofloc, we are still on the experimentation stage for that in mudfish. And a very good story of that biofloc system is with Dr. Chris Kaipang in the larval rearing of Pineus vaname, I think, if I'm not mistaken. So it's a good, I think they have already produced some manual guide or field guide for that, and it's easy to, maybe you can contact Dr. Chris for that. Thank you.
Thank you. And another, a few more questions for Dr. Cass. Have you done this induced spawning also in catfish, and how does it differ? And how about in other fishes?
Wow, very good question. Because it's totally different from catfish. In catfish, you really sacrifice the male. So it's very, you have to take the pituitaries and you take the sperms or the of the gametes of the catfish. So and then fertilized, and that's why it's a simplistic model in the log or in our mudfish, because we just inject them in hormone and then we place them in hapa nets. No, actually we close. Usually we cover them with a black cloth in fiber. If we use the fiberglass tanks, we usually cover them with black cloth, because maybe the characteristics of mudfish is lights off, so we usually cover them. It's not lights on, but it's lights off, so we usually cover them with the black cloth. So it's totally different, unlike in catfish that we have to sacrifice the male, get the pituitary glands, to induce into females, and then for the sperm we usually do the surgery or open the part of the catfish and do the IVF. So it's totally different. So that's the behind of different techniques in fish now in the spawning.
Okay, thank you so much. Now we have a question again for Dr. Tulin, and this is from our beloved Professor Dr. Violeta Villegas. Is BioMEG ready for adoption or pick up by the private sector for greater distribution? Have you done some partnership or conversations or discussions with any industry takers, perhaps?
Not yet, actually, for big industry, but I was actually planning to do a wider field application. The DA actually contacted me to do experiment in the Zambales area, the Lahar area, actually, to be planted with purple yam. I was actually excited in that kind of arrangement, but maybe we will push through with that. But as far as industry is concerned, not yet. But I think we are moving there, because we have already our LGU, actually, for one, and the neighboring LGUs, we have conducted already training. And in fact, there is also an interest in some of the farmers to utilize their land actually for sweet potato production. No? Because in our area, for example, in Leyte, we have actually a sweet potato processing plant. So we plant sweet potato, and then these farmers are actually also encouraged to plant sweet potato, because there is already market for that. And so we apply this technology. They have their lands, and I think that negotiation is also ongoing. But as far as big commercial industry is concerned, not yet.
I think thank you so much, Dr. Tulin. I think that question is also for Dr. Choresca. Have there been any interest from the private sector in using your technology and adopting, or for a wide-scale preparation of the spawns?
Yes, that's a very good question. Actually, recently, we received many queries regarding how to collaborate with them in our stakeholders, especially for private farms, in order to assess them, especially for the market of this of this mudfish. So actually, we are still in the infant stage, and for the hatchery production, I believe now we only rely on the wild, but with our technology, and they hear this good news already that we are starting the induced breeding, and anytime we can get our fingerlings and fry, and now they are we have lists of stakeholders who are willing to venture also in aquaculture technology in mudfish. Actually, we are just starting our grow out experiments, and now we, based on our trainings conducted, they are positive in because this mudfish is our if our traditional fish, should I say. Many are looking forward for this fish to revive, since the soup is very good according to our grandfathers and forefathers. So actually, we did some surveys, and usually age 40 up, they knew this. We have this haluan or our dalag because of the pesang dalag, and our new generations, usually they don't even identify our mudfish. So this is really, an intervention. And of course, in aquaculture industry, the aquaculture, actually we are promoting for the aquaculture and for conservation also of this fish, because as I've said, it's still dwindling. And the responses are positive, should I say. And now we are having our schedule in producing this fish, and our promotion of this technology is ongoing. Hopefully, we can, because we already have here in Luzon and in Visayas, and hopefully we can extend in Mindanao, because they also wanted to have this kind of technology, especially in Maguindanao, in North Cotabato, and because this is the legacy in Mindanao. And hopefully the DA Biotechnology program will finance again and funds our projects for the promotion of aquaculture technology in mudfish. Thank you.
Dr. Mandala is here, so he can hear you.
Yeah,
a few more
actually,
questions.
Dr. Bong witnessed those fisher folks wanted to have this kind of technology. Thank you.
Thank you. Now, let's go back to Dr. Tulin. So there are two questions that I want to raise. The first one is on the isolation of the Bacillus megatherium, since it is endogenous in our soil. So, for example, when you are isolating it, how much soil do you have to get in order to harvest a significant Bacillus megatherium? And then the next one is, can this be used in hydroponic system?
Okay. The the production or the cultivation of the pure stock of Bacillus, of course, follows a standard procedure, of cultivating microorganism. Well, you only need one spore in order to cultivate many, but basically, you can mass produce it. What I'm saying is you can mass produce it using the substrates that we actually specify. But during the isolation, of course, this isolation, you really have to characterize, and you really have to get the pure clone or the single clone, and before doing that. But in terms of mass production, I think we have already the technology for that, as I was discussing or describing in my presentation, using organic wastes. So it's all there. Then regarding the use in hydroponics, actually that's also an intriguing question, because this is a soil enriching. I don't know, but if you can microorganism, but if you can do that in a soilless room, let us try and see if it can actually, from a kind of water source. But yeah, I'm intrigued with that question. So I tried to find out. Thank you for that question.
Okay, thank you, sir. Okay, and now we're moving back and forth because we need to accommodate a lot of questions from both And so one of the questions that is very interesting is how many times, this is for Dr. Choresca, how many times do you have to inject the hormones? And does it depend on the weight of the breeder or the male? Does induced spawning applicable in shrimp? And I think that's it for now. So if it's applicable for shrimp, and if, oh, there's another one. We have tried induced spawning in catfish with overprim. Is it also possible to use it in mudfish? Okay. Right.
Wow, very interesting. Yeah, we only have once for the injection. Usually we had our follow-up injection the next day if they don't spawn. And then usually we check the fish. If the fish is fully mature, then we just only injected it once, because if you inject it, if you don't check the fish, so it's your your hormones is expensive, and you just inject them twice or many times, and it's a waste. And then we only had that once, but we had follow-up sometimes in the next day in order to confirm the maturity of our fish. And then, yeah, overprim. We had a series of injections and experiments on commercial hormones like HCG, LHRHA, and others. So we tried that one and we optimized, and then we compared that also with our developed pituitary gland hormones, from other fish. So I cannot disclose yet. So we have tried those inducing hormones, and then for crossbreeding of the catfish, yes, you can have our native and crossbreed with other African catfish. And yes, it's different from fish to shrimp. For shrimp, we used to mature them using ablation of the eyes stalk of the shrimp. So that's totally different from catfish. And for catfish and other fish or mudfish. So I think that I missed something, or
it's okay. You were able to respond to all. Thank you so much.
Okay, thank you.
Yeah. So we still have a lot of time. So get your questions in. And so there are other questions for Dr. Tulin, and one of those is are there environmental conditions or factors that could possibly affect the growth performance of BioMEG? And again, from Dr. Villegas, with the high cost of fertilizers, can application of BioMEG possibly reduce the rate of fertilization without sacrificing yield and quality? And yeah, problematic soils. Is there an ongoing study on BioMEG performance on problematic soil and problematic amount of rainfall? Because of course we have dry spells, and since microorganism needs moisture to proliferate. So those are the three questions.
Yes. Environmental factors affecting activity, or the effectivity of the microorganism. I would say that, of course, you were saying rainfall or moisture. So if it's flooding, if there is flooding, I think there's a probability that you will not have a good performance of the microorganism, because if it's flooded, I think microorganisms will also be washed out. It might repopulate later on, but that's quite also difficult to trace. No. We have not done any experiment yet on moisture level or moisture or drought, but as long as there is moisture, because we base on idea, I think you're right. Activity of microorganism actually also depends on the moisture of the soil. So it needs also a critical moisture in order to function. So I guess if it's really severe drought, if there is, then I think we can work on that. I'm also interested to work on that. And I think maybe because actually we have already studied the viability of the microorganisms, and to tell you the microorganism is able to survive for a long time, as long as that's why we choose a microorganism that is able to sporulate, because they can actually become exist as spores and then revive when the proper conditions like proper moisture in the soil is there. So we store them as spores, as active spores, and then revive them in the soil when the soil is ready for, so we don't actually apply as live microorganisms, but as spores. So that is something that we can also try to monitor in the field. Thank you. What's the
Okay.
other question, Mamola? Is that?
Yes. The other question is on the cost, because we have now the cost of fertilizer is going up, as you know. So this will this the application of this technology will lower down the use of fertilizer.
Yeah,
So
when you talk about the economies of scale, the cost of that can be reduced. I was saying 218 pesos per kilogram, but if you actually produce or mass produce it in larger scale, I think you can actually reduce that cost. In terms of the use, because you use also complete fertilizer, because the recommended rate of complete fertilizer is also we have established in some plants, but try reducing this waste. It's also very tricky, because try reducing the recommendation of the complete fertilizer will also give you possibly a different effect. So there's also a balance on that.
okay. Yeah. Yeah. Okay, so I think this would be the last question for Dr. Choresca, and then I'm going to ask one question for you both. Dr. Choresca, I would like to know, since African catfish is non-native and usually it is commonly reared due to its commercial value, especially here in Luzon, may I know if promotion of native species is our priority, or are there certain regions in the country that favors the culture of native fish?
Yeah, that will be our priority. And hopefully by this year or next year, our program for the DE Biotech office is on the indigenous species. So we are reviving our native catfish, and hopefully, actually, in Luzon, they are now producing this African catfish in anywhere, But due to the dwindling stocks of our native catfish, so it's difficult now to, for broodstock collection. However, we had identified already from North Cotabato and also in Maguindanao, and we have that some areas. We have a collaboration actually with the University of Mindanao State University in Maguindanao for the collection of this our native catfish, and also we are trying to reach out in Palawan and Zambales. And one of our projects in ATBI, so for that business incubation, is the promotion of this our native catfish. So part of the project actually that we had discussed during our MONCOM with Dr. Bong is the revival of this native fish, especially like our pigik, our ayungin, and climbing person, of course, our dalag. So it will revive our, cultural and indigenous species that we had before. And we can see this, before we can see this in our rivers and everywhere, but now it's really, dwindling in our areas. And the price now of this indigenous species is quite high compared to our some of our fish commodities. So I think it's a very good question. So I hope that this will be prioritized in the field of research. So in our effort here in FBC, one of our commodity roadmaps is on the indigenous species. So hopefully it will be funded soon, and our stocks will be revived, especially on this native fish. Thank you.
Okay, so my last question, which will be answered by you both, will be on whenever they need information on BioMEG, where can you be contacted? And also, if you are amenable to giving trainings and in many different parts of the world, because I know we have international participants here who may be interested in your technology. both you, BioMEG, and also the spawning. And what would be your last message on encouraging a Pinoy biotech for our researchers and our general public? You may start, Dr. Tulin.
Okay. First of all, yeah, there is a need, or if somebody's interested to go to avail of training or anything related to our project, the Biotech Biotechnology Program Office can also be reached. And of course, our university can also be reached through our extension office. We have conducted trainings already to our LGU and other regions of and other places in the region. And we have produced, of course, IEC materials, including a demonstration video for this purpose. No? And yeah, my message is, of course, I congratulate Ola for this hosting this event and for enabling us to share this information to our Pinoy biotech enthusiasts and would-be researchers. I really encourage our young scientists to go on Pinoy biotech, even for the conventional. Perhaps we can move into the more modern and then later on, subject to our compliance of some regulations, which I think Ma'am Ola is very aware. And I'm very glad that we have a number of researchers in biotechnology, especially crop biotechnology. Thank you so much.
Thank you so much, Dr. Tulin. Now let's have you, Dr. Choresca, please.
Yeah, actually, we are very happy to share with you our technology. And in fish, it's quite infant, and we are still on growing stage. And I know crops and of course livestock is already on the limelight. And in our case, you can reach out here at the Science City of Muñoz. We are inside the BFAR, National Freshwater Technology Station, BFAR in FFTC. And you can contact also ED4DI and DA Biotechnology Program Office. Actually, we were once reached out using the announcement of the DA BPO, and I believe this time, Tita Ola, it will go international. So thank you very much for the promotion of this kind of webinar, especially our biotech products, especially in fish. And we are happy now. We are reaching LGUs, farmers, and then we are reaching out regional. So now we are in Bacolod, in Guimaras, in Iloilo, here in Central Luzon. And I hope we can reach in other parts of in the Philippines. And I really wish to thank, of course, the ABPO for this funding. Actually, this is just an ambush funding, and we have what do you call this? Initial work that we through Director Ann before that I lobbied this kind of technology, and this really helps for I said to her that it really helps to our fish farmers. So now I'm lobbying for the fish in a cup, because usually we give sardines, right, to our disaster-stricken areas. So I hope somebody also to fund our mudfish in a cup. So we used to translate that in a noodle type. You can see that in other emergency or in convenience stores, that you just put hot water. So it's the same thing. So I'm looking for the funding on metabolomics of this mudfish, so that we can support the knowledge of our forefathers that this fish is very good after birth of women. So thank you very much for this opportunity. Thank you
So much, Dr. Choresca and Dr. Tulin. Let's give them a virtual round of applause, everyone. Now, before we close, I'd like to invite everyone, all the participants, to please look at the chat box. We have the link to the evaluation form. It's a post-webinar survey, and this will be your ticket to get your certificates. And we also would like to improve our webinars, so we value that you respond to this webinar survey, post-webinar survey. So thank you once again. Please don't leave yet. I'd like to introduce to you the person who's going to give the closing remarks, our Director of the Philippine Agriculture and Fisheries Biotechnology Program, or DABPO, the Department of Agriculture, the chair of the technical working group for the drafting of the Joint Department Circular for Genetically Modified Animals and Animal Products Rules and Regulations. Let's welcome Dr. Claro Mengala. You have now the floor, Dr. Bong. Where are you? You're in a car? Okay. Yes. You're frozen.
Internal
Okay.
budget hearing in Pampanga. Hello, am I
Yes,
clear?
yes. You're okay.
Okay. Yeah, thank you very much, Dr. Ola. I would like to express my sincere appreciation to ISAAA, of course, for the unwavering support to promote Pinoy developed biotechnologies by our own scientists for the benefit of our farmers and fisher folks. Actually, when we discussed about the DA Biotechnology Program Office during the budget hearing, we were able to express our intention to continuously expand the program. And we are hoping that because of the presentations that have been made by the DA BPO and the Bureau of Agricultural Research, of course, additional funding might be possible for the program. And of course, because of the possibility of this, we will be able to fund more projects and programs. So webinars such as this plays a vital role in disseminating information on the from research and development initiatives of the Department of Agriculture. It also provides platform for discussion among you, the attendees, of course, the participants of this important seminar, and the scientists or technology developers to have a better appreciation and understanding of the product that were developed using biotechnology tools. Both products that were highlighted in today's webinar are being supported by the DA Biotech Program Office with the aim of increasing the yield and enhancing the nutritional quality of our root crops, as well as improving the spawning technology for mudfish production using traditional biotechnology approaches. By supporting these products, the Department of Agriculture Biotechnology Program Office is helping drive innovation in the agricultural sector and promote sustainable practices that will benefit farmers, fisher folks, and consumer alike. Moreover, the use of traditional biotechnology approaches for mudfish production is an excellent example on how biotech can be utilized to improve the utilization of biotechnology tools in the agricultural sectors, as well as the research and development programs and initiatives of the government, particularly of the Department of Agriculture. By participating in this webinar, you have gained knowledge about the latest biotech innovations and the role of biotechnology in the agricultural sector. Once again, thank you very much for your participation. Thank you very much, ISAAA. And thank you very much to our two experts, presenters, President Tulin, and of course, Dr. Choresca. And I hope that this webinar has been a valuable learning experience for all of us. Thank you very much. Mabuhay po tayong lahat. Good afternoon.
Thank you very much, Dr. Bong. And that ends our webinar. Thank you so much again, Dr. Tulin, President Tulin, and Dr. Choresca. At this point, we would like to invite everyone to our next webinar, which is scheduled on March 31. It's going to be on aquaculture, fishery, biotechnology, and this is going to be an international one with Dr. Eric Hallerman from Virginia Tech and Dr. Muji Santos, again, from NFRDI, our academician. And we hope that you will come with us and see what's happening globally in terms of aquaculture and fishery. Thank you very much. And I hope that you will join us again. And thank you. Good afternoon po. Bye-bye.
Thank you. Bye-bye.
Bye-bye.
Thank you po. Bye-bye.
Bye-bye. Thank you so much.
Bye-bye.
Bye.